Hello, everyone. Thank you for joining our webinar again, Advancing Clinical Research Through Targeted and Full-Length Single-Cell Sequencing. So again, my name is Irene and I'll be your host for today. Our speaker today is Dr. Sujata, Singleron's Regional Application Manager. Just before we start the session, if you have any questions, you can definitely use the Q&A function at the bottom of your Zoom window, and we will address all the Q&A questions after Dr. Sujata's presentation. And without further ado, let's invite Dr. Sujata.
Thanks, Irene. Okay. Good morning, good afternoon, good evening, everyone, depending on where you're logging into. I'm Sujata. I'm the Regional Applications Manager at Singleron APAC. And for today's session, my aim would be to share sort of different ways how clinical research can be advanced using single-cell technologies, and more specifically through targeted and full-length options that are available. Just to kind of get started, we all know that you can do bulk RNA sequencing or single cell RNA sequencing. So what's sort of the difference? If you were doing bulk analysis of a sample, say a tumor sample, a cancer sample, the data that you get would be an average information of that sample. So here you can see it's an averaged clone information. So, and it would mostly be the most common clone in that sample. And so if you were sort of using this in a clinical setting, you would think of the known drug to target that based on this information. And then what remains would be the other different cell types or clones that kind of escape this drug. And so what single cell data would give you is information of all the different cells or clones that were in that sample, and any sort of biomarker signals from that, which means you can actually go in and find more precise drugs to help treat that condition. And from there, you can really build on it. You could actually use single cell to kind of profile. So you're monitoring a sample and then or a tumor, and you only intervene when needed, when you see that there is a sudden increase in a certain clone. And then based on sort of the biomarkers and information that you have, you can do a pre-treatment analysis and a pro-treatment analysis to understand at which point, what was sort of the cells that started off, where can you sort of improve your detection of that particular sample. So overall, in sort of clinical applications, it can be sort of in the research space. So you're doing clinical samples and you're doing sort of answering questions based on just developing tissue or disease tissue atlases. You're identifying pathways, looking to develop drugs, understanding biomarkers, looking at developmental biology, and then it can then move slowly into clinical practice. So here you're actually treating sort of tumors, infectious diseases, diseases of tissues, etc. So it sort of straddles both these, and single cell applications can be in any of these fields or even at the junction of these. So just sort of to kind of understand what a single cell workflow, what do I mean by that? So that means you kind of start from a tissue, you get a single cell suspension, and then you actually have to do your assay to capture your single cell information, you sequence, and then you analyze your data. So your options would be, how would you capture your cells? So we have from Singleron a microwell-based technology. So we use chip to capture your different cells individually from a sample. And then we use a barcoding bead to capture the mRNA. So mRNA are mostly polyadenylated. So you have a poly A tail. So we have a bead which has unique barcodes and a poly T that will capture your mRNA. So this is sort of standard single cell platforms do an mRNA profiling using poly-T. And we do know now that there are signals that can be missed when you do just a poly-A capture. And many of these are extremely important if you are in the clinical space. So for example, just by the word, anything that doesn't have poly-A will not be captured. So for example, those are viral and bacterial signals, because they inherently don't have a poly-A in their mRNA. And then you also have other markers, cancer and disease markers, such as mutations, as SNP, gene fusions, splice isoforms. So you need information of all of this before you can actually figure out what sort of treatment or how is the disease developing or detecting. So all of these things are required, and mostly these don't get captured in basic poly-A capture data sets. You could be needing to look for a very rare or low expression transcript. Again, this could be missed. And then finally, if you're looking for immune markers, you need to know what T cell or B cell receptors are. These can also be missed if you were just capturing your mRNA based on poly-A. And so we do have an array of other kits and assays that can help you capture various different layers of data other than just using a poly-DT capture. And for today, I'll focus on two assays, one that helps pick up SNVs or gene fusions, because these are important in sort of the clinical setup, and then one that can also look at full-length coverage. This is where you capture more of the length of a particular transcript. So you understand, you can capture or detect all the different splice forms, splice sites, etc., all of which are extremely important in the clinical research. Right. So to leverage single cell sequencing in clinical applications, it's a really broad field. So for the purposes of today's webinar, we will try to cover it under these sections. One is to just see how single cell helps understand tumor biology, and then how that leads to some biomarker discovery, how you can use it for early detection of disease, then pathogen detection, and then finally just a few examples on how single cells can be used in the translational clinical research space. Right, so I've kind of put tumor biology and biodiscovery marker together, so we kind of can see how you need to understand one before you find the other, and then we can use this example. So this is sort of a study that was looking at the tumor heterogeneity and the microenvironment of advanced non-small cell lung cancer. And so this scenario, this disease has two subtypes. So that's adenocarcinoma and squamous cell carcinoma. So these two are sort of histopathology distinctions, but there are no reliable predictive biomarkers or biomarkers. So the authors decided to comprehensively profile the tumor microenvironment and then kind of get that data so you can actually make a biomarker discovery. And then once you have biomarkers, you can see kind of interesting biomarkers in the data set, you can then decide how you want to do your patient stratification and then improve personalized treatment. So all of this kind of goes together, and depending on which stage of the research you are. And so for starters, the study has kind of, they have 42 patient samples. So this is sort of the different patients from the different subtypes, that adenocarcinoma, squamous cell carcinoma, and then others that cannot definitely be kind of identified into either of these subtypes. So you can first build a whole patient, sort of the disease atlas for based on all of these patients. So you have the different cell types, how their composition changes across the different conditions, etc. And then what you can do based on this is see if you are able to kind of come up with markers. So what this study kind of found is that within the lung squamous carcinoma subtype, there is a very high inter and intra heterogeneity compared to the other subtypes. So there's already interesting markers there that kind of tell you that you need to look into the different cell states. And then they also go in and look at the, other than the tumor subtypes, they also look at the composition of the immune cells. So there's T cells that they sampled, which I've not shown here. But then if you look at the second sort of figure here, they actually looked into the correlation of the cellular components, tumor, and the intertumor heterogeneity. And they actually find in one cell type, you see neutrophils of high proportion. So they kind of concluded that in this disease, neutrophils seem to play a very important role when compared to the T cell response that they found. So both knowing the cell types, the subtypes in the sample, as well as sort of the heterogeneity within the sample, help them kind of understand the immune landscape. So here you have biomarkers possibly from cell types and immune landscape. And then finally, you can kind of do all of your analysis and then also see if there are signaling biomarkers. So here we are looking at sort of how cells signal each other. So for example, here for a particular subtype, for the squamous carcinoma subtype, FGP pathways between sort of stromal and the tumor cells are supposed to be activated. So that was sort of a hallmark that's considered. But this study kind of shows evidence that that is not really true for all of the samples that they studied. So you clearly need to have some kind of patient stratification before you go in and just treat the particular pathway using drugs for just this particular pathway. And so all of these insights into the particular disease comes about when you sort of in-depth single cell sequence the sample and then kind of understand both the cancer cell types, the immune profile, and all of the sort of cell signaling between these types. So this is sort of discovery based. So you're just kind of digging into the different sort of cancer tissues and figuring out what are the biomarkers. You can also use single cell for validation. So here's sort of a study that already has come up with biomarkers, but they did it based on proteins. But at the end of the study, before they move on to actual implementation, they need to do a single cell sequencing validation. So here they were studying blood cancer, and so they're looking at some sort of biomarker for early diagnosis. And so they first find protein biomarkers, and based on that, they do a single cell sequencing of bladder and adjacent tissue to make sure that they can confirm there is sort of transcriptome or mRNA expression of these markers at the source tissue. So keeping in sort of a track with early detection of disease, single cell, again, can be used for understanding or sort of looking for early signals of particular diseases. So the example I'm going to use here is clonal hematopoiesis of indeterminate potential. So this is also known as CHIP. So this is a sort of condition which involves age-related accumulation of somatic mutations. So it's mostly seen in the hematopoietic stem cells. So with age, you kind of accumulate these mutations, and then suddenly there's a sort of clonal expansion. And then at that point, data have shown that this associates increased risk with both hematologic malignancies as well as a lot of other age-related conditions. So for example, heart conditions, cardiovascular, diabetes, etc. So this is sort of an early signal, and if detected early, could kind of prompt early intervention. So for CHIP, we kind of know there are these particular genes that are involved, and it's not just sort of the genes. We also understand there are actually particular mutations in the genes. So if it was just the genes, you can just do a general capture and you might be able to, you can detect the presence or absence of that gene. But what we actually need is more in-depth information on common mutations that are implicated in CHIP. So based on the genes, these are sort of the known mutations. So you need an assay that can actually target these directly. And is there a need for that? So just some data to show if you use just sort of capture with poly-DT, you find a certain number of cells that have this particular mutation. So this is data for R248Q. But the moment you put in specific probes or like a targeted panel, you see that way more cells actually have that mutation. So you're kind of missing about 40 to 60% of the information if you didn't use a more targeted panel when you're trying to do such early disease detection. And so just coming back, if you've kind of got a PBMC sample, you can run one of these panels and know if there's any indication of CHIP. And as I mentioned, there's different sort of risk factors, including hematologic malignancies. So you can go from knowing if there is a sort of early disease detection to actually sort of targeting and confirming if you're seeing markers for that particular disease. So for example, you can use a blood cancer panel that we have that has mutations or gene fusions that are known in blood cancer. And so as these authors have done, they've actually studied two different hematologic malignancies, MDS and AML. And so they wanted to see which were the different cell types that tend to have these mutations. So here they are studying IDH and KRAS. So these are known SNVs in blood cancers. And so what single cell helped them do is sort of come up with all the different cell types in each of the different samples, and then from there you assign it. You can assign what's the percentage number of cells that actually have this mutation and how strong is the expression. So this is sort of information that you get by screening through a blood cancer panel. And from there, you can go in and actually do pathway analysis. So here is sort of cells, so erythroid cells for these two mutations. And at the end of the study, not only did it become obvious that the samples or the disease is quite heterogeneous, and because of this sort of enrichment pathway analysis, they were able to find out that there is sort of a resistance probability to chemotherapy, which is quite high because of the ferroptosis pathway being active. And so they were able to propose some novel target treatment for these conditions. Right, so these are sort of kits that are available off the shelf, but say you have another sort of disease condition or tumor or cancer that you are interested in studying, you can actually put together one of these panels. We can help sort of do that for you. So you find 10 markers and then we can help put that together and you can do the same kind of screening. So a kind of FocuSCOPE or targeted panel approach, say you were interested in looking at a particular gene, so you want to screen these genes for SNVs, but at the same time, you're interested in particular mutations. So you can come up with sort of a result where you're seeing your mutations being present. So these dots mean these are positions where you have these mutations. But what if I want to know more about this? So I want to know the point where the mutation is. At the same time, I also want to know the full transcript information, which you wouldn't get if you were just using a targeted panel. It would be more a presence absence. So for that, you can use a full-length transcript detection kit. The one we have is called MobiuSCOPE. So what this gives you is for that particular gene, not only do you know sort of the places in which the mutations are there, you can actually find out all the different transcripts that are, you can know all of the different transcript detected for that gene. And that gives you more sort of information on how they are expressed. So what are the transcripts you're getting? How is the splicing? How is the sort of alternate splicing? Where are the splice junctions? So just sort of covering more of the transcript gives you more information on the markers that you are interested in for that particular disease. And that for sure will give you sort of more information. So this is sort of visual representation of what it would look like if you were trying to look across the transcript. So say, for example, you have gene C and you have mutations that you want to detect with FocuSCOPE, you'd be able to just tell where those mutations are. But if you actually ran a full-length assay, you can actually line up your transcripts on the gene and kind of see what are the different splice junctions where these alternate splicing happens, etc. And all of these do play very important role in diseases and hence in clinical research. So we were talking about mutations. So those are point mutations, deletions, insertions. There are other diseases where you actually need to look for fusion genes. So for example, CML, where the detection of the presence of these fusion genes are important. And so you have an option to actually do a panel where you can detect the presence of fusion genes of interest. And so that would look like presence absence of these particular fusions. Or you can actually look at the transcripts. So if you're interested in getting more information, you can do full-length transcript sequencing for the same two fusion genes will give you more information and more robust data on the detection of these fusion genes. Right. So this was more about how do you kind of do early intervention? What are the signals you get of diseases, or how do you kind of find markers in diseases? The next part is on pathogen detection. So this is more on how disease mechanisms can be detected if you were looking for pathogens. So in an infection situation, there's two ways you can do single cell work. One is you can actually study the host response. So you're actually working on the tissue, trying to understand what all changes in response to infection or a pathogen. Or you can actually try to turn it around and study the pathogen. You can try to see how where is the presence of the pathogen? Can you actually detect it in the cells, etc.? So either of these are approaches that are used in the clinical research, and each of it gives a different sort of information, and you can do that something like this. So in this study, the authors are looking at pediatric EB virus associated HL through single cell. So here they're interested in the Epstein-Barr virus infection in the HL condition. But the whole study actually just looks at how the host cells respond. So you just need a normal single cell assay that captures polyase like check-scope to actually just sample all the different cell types and then tell you what are the different pathways that are activated, etc. But if I was interested in actually studying the presence of the EBV, you can actually come up with a panel. So here we have an Epstein-Barr virus panel. These have markers that that can detect presence of the EBV virus because they are genes of the virus. So here, for this particular study, they are trying to understand where does the disease originate in sort of chronic active EBV disease. So they need to sort of know where is the virus present. So they need to do a viral profiling and then from there sort of figure out. So here the basic single cell will give you the different cell types. And instead of actually going into the cell types and trying to understand their response, they actually use the EBV panel to detect which are the different cell types that actually have EBV infection. So you can see pre-treatment, they concluded that right from the hematopoietic stem cells, there is EBV infection present that's sort of there across all of the other cell types. And so it kind of gave them an option to kind of try an innovative therapy strategy. So what if you sort of replace the hematopoietic stem cells by transplantation? And they do find sort of in a lab setting that if you do that, you can see that the EBV infection drastically has reduced or gone. So this is sort of a setup where the viral signals are used to detect the viral signals and then come up with a therapeutic strategy. And so based on this sort of principle, you can kind of develop a custom kit based on the pathogen of interest. So even if your sort of infections, viral or bacterial, doesn't have a poly-A tail, which is not captured in standard single cell platforms, you can come up with sort of a custom kit. What you'd need to know is sort of markers on the virus that can be put on probes and then you can capture. So here's an sort of publication based on Zika virus. So here they're trying to quantify Zika virus in sort of in the sample. And you can see that you can do both your basic single cell to find out all the different cell types and then also kind of see what is your Zika virus sort of presence in sort of a Zika infected sample. So this is sort of an additional information that helps you sort of, gives you information on the viral signals. And this can be adapted to any other sort of pathogen. We can also sort of, you can also do this for bacterial signals, more like intracellular bacteria, ones that have already sort of integrated into the cell. And so this is again an area that's becoming quite popular where cancer cells and depending on sort of its coexistence with intracellular bacteria being studied. Right. So coming to sort of the last stretch, this is where translational clinical research. So all of this can be quite sort of on the lab side of things, more research biological questions, but you can also bring it more to a clinical research setting. And so sort of one application that is quite that's quite popular now would be CAR T therapy. So if you think of the whole cycle of any kind of sort of administrating therapy, monitoring and treatment, you have to work with many different sample types like PBMCs, biopsies, organoids, engineered products. So, and in each stage, starting from detecting sort of tumor and then coming up with a therapy, then infusing it back, monitoring and carrying on all of these steps require a single cell sequencing to understand. So in terms of sort of a example, this is study that's looking at engineered T cells. So that's CAR-T study, and so the authors have sort of come up with this CAR-T, and they needed to check if these are actually having long-term persistence in humans. So then they kind of do a study where they've infused it and then sort of use single cell technology to make sure that these cells are actually sort of surviving a long term for long term, so that they are persistent and there's functionality of the T cell. So this is kind of a application where single cell is used for the whole CAR-T process. And so just before we end, I'm just like flashing different studies that have used single cell work in sort of clinical research. Say you're sort of working on ovarian cancer, you want to understand if the Treg cells are actually sort of inhibiting your chemotherapy, or if you want to see how the different trajectories are or novel biomarkers can be detected in diseases such as primary neuroblastoma. So you need single cell work to do that, or just understanding sort of the landscape of host responses in diseases such as COVID. So you can run like longitudinal clinical tests using single cell work. So these are all sort of translational clinical studies that are kind of showcase how you can use single cell for both and give you ideas on how to think about using single cell in clinical applications. So just to kind of revise here, these are sort of the assays that we have that can help you, as I mentioned, not just do mRNA profiling to build sort of basic single cell atlases of disease. You can then use various other assays to go in depth to kind of find biomarkers, understand pathogen work. We can also do immune profiling, which I've not gone in depth. So you can take any of these scenarios where you're interested in then understanding the TCR or BCR profiles in the samples. And then there's also options to actually do ATAC-seq. So that's to understand chromatin accessibility and what roles they play in disease. So there are a lot of single cell assays available. That's more than just doing poly A capture. And so these are all things to think about when using clinical applications. So just a little bit on what Singleron can provide. So we have instrument-free, easy to load scope chip assays. So all of the different assays that I spoke about are available. But we also have walk-away automation if you want to do high throughput samples. We do microvell capture, so you can capture between 20,000 to 30,000 cells depending on your requirements. And if you do work with cell sizes that are larger than the standard 40 microns that most single cell platforms work with, we do have a chip for that as well. We also do tissue dissociation, which is sort of upstream of your single cell work. If you're working with tissues, you do need to dissociate those to a high quality single cell suspension with very high viability. So we do have sort of automated dissociators for that. If any of these assays are of interest, we are present worldwide. So in Singapore, our team is from Singapore, but we do have offices in Europe, US, and China. And then we have our trusted distributors in the other places. Please, in case you want to try any of the assays. And also we do have a service labs in all of our locations. So we do end-to-end services. You can submit your sample and we run it through the entire workflow with basic data analysis. And so these kind of, we can sort of advise you on the right assay to use based on your sort of clinical application. And then you're able to just send us the sample and receive your data back. That's kind of what I have for today. I hope it has been helpful to kind of plan your clinical projects or the applications and how single cell can you can leverage single cell for those. Thank you.
I guess that's pretty much, we are getting to the end of the webinar. There's no further questions. I hope that all the attendees, you guys have a very good session with us. If you have any more questions on single cell sequencing, do contact us at sales.sg@singleron.bio. After this webinar, I will also send out an email to all the attendees so that in case you have any questions, feel free to just reply from the email itself. Yeah, so that pretty much summarized the whole webinar. Thank you everyone for joining today, and I'll see you at our next event. Thank you.
Thank you.